While we wait and till people get into the room, my name is Oliver Fien. I'm director of the West Coast Metabolomics Center. I would like to welcome today this month's speaker, Matej Orešič from Örebro University in Finland, right? Or is it Denmark?
Well, that is in Sweden. I'm also in Finland.
Oh, Sweden.
Yeah.
Oh, there you go.
Yeah.
It's Nordic. Sorry. Dr. Orešič holds a PhD in molecular biology and genetics that he got from his time at Cornell University, where he was 1992 to 1999. He has initiated the popular MZMine open source project with the primary publication in 2006. But also subsequent publications in 2010 and in 2023, which is now MZMine 3. As of 2016, Professor Orešič was made a lifetime honorary fellow of the Metabolomics Society, and he's a co-founder and former chair of the board of the Nordic Metabolomics Society in Europe. He was also, in his career, he was head of computational biology and modeling at Beyond Genomics in Waltham, Massachusetts, and he was a bioinformatician at Lion Bioscience Research in Cambridge, Massachusetts. From 2003 to 2014, he was at VTT Technical Research Center of Finland in Espoo. 2014 to 2016, he was the principal investigator at Steno Diabetes Center in Denmark. And 26 to 29, a group leader in systems medicine at Turku Center of Biotechnology, University of Turku. And from 2017 to present, he's a professor in medicine with a specialization in systems medicine at Örebro University. He is still at Turku, if I understand. So it's 2 appointments, right, Matej?
Yes.
Okay, so 2, both Turku Center for Biotech and at Örebro. So with that, I welcome our speaker, Matej Orešič. He will talk about the metabolome in non-alcoholic fatty liver disease pathways, metabolic models, and biomarkers. Dr. Orešič, the floor is yours.
Thank you. So I will start with acknowledgements from the start. So basically, of course, any work that we do is collaboration. So I would like to thank my team and particularly clinical collaborators such as Hannele Järvinen from Helsinki University, who has done work on the liver studies that I will mention, and then the European projects, EPOS and LITMUS, that have allowed us to do the work on the clinical samples across the Europe on the fatty liver disease, and then also Jennifer Schlesinger and Thomas Webster from Boston, who have done the work on the PPAR alpha humanized mouse model that was used in exposure studies that I will talk about. So what I will talk about here is some background on the fatty liver disease, and then highlight the heterogeneity. So it's not only about fat, but also what kind of fat we have. And then talk about metabolome across the full spectrum, modeling also metabolism of hepatic metabolism in fatty liver disease using genome scale metabolic model, and then also look at the potential role of environmental contaminants in the progression of fatty liver disease. So non-alcoholic fatty liver disease is a broad spectrum of conditions that range from obese steatosis, essentially accumulation of the lipid droplets and lipids in the liver and hepatocytes, and then which can progress towards the more inflammatory condition, which is called non-alcoholic steatohepatitis. And then if it reaches a fibrotic state, there are several grades of that, eventually can reach to cirrhosis and possibly also leads to the liver cancer, hepatocellular carcinoma, which is currently, because of obesity being more and more prevalent, the NAFLD is currently the main cause of the liver cancer. And on the other hand, also fatty liver is associated with insulin resistance, metabolic syndrome, and the risk of type 2 diabetes. So it is in the intersection of many possible diseases. So that is, for that reason, this is being a very important public health problem. And there are three main overlapping views of NAFLD. That one is steatosis, which is accumulation of fat in the liver. So that's basically meaning that you have lipids in the liver that are being accumulated, and usually these lipids, like triglycerides, are being stored in the lipid droplets. But that by itself is not necessarily a pathogenic condition, but it can lead to that. So fibrosis is then a more progressive stage of fatty liver, where is a accumulation of extracellular matrix proteins, including collagen, that occur over time and lead to inflammation that leads to excessive amount of scar tissue that builds in the organ. So there are several stages of fibrosis. So the hepatologists usually use like from 0 to 4, 0 being no fibrosis, and then 4 is being a cirrhosis, the final stage of the fibrotic stage. So non-alcoholic steatohepatitis is sort of in between, or NASH. So that's a steatosis which progresses to inflammation and liver damage, which will then lead to fibrosis. So that's sort of like a overlapping condition, but it's very commonly today referred to that is overlapping with fibrosis, but it is more or less like a grade stage 2 to 3 fibrosis where the NASH is being diagnosed, but it is not officially like a disease. It is sort of a condition that is being used for clinical trials and in the research, but it's not officially diagnosed as a disease. So fibrosis, steatosis are the official conditions. So the gold standard for diagnosis these conditions has histology, meaning that you have a biopsy. Now, of course, in a regular public health setting, you will not get a liver biopsy for just looking at the liver fat. So it only occurs when there is a severe form of fatty liver disease is suspected, like cirrhosis. So there is currently no actual way to really diagnose the stage of the fatty liver disease. So there is unmet need, and that is the reason why there is so much interest currently in prognostic and diagnostic markers of staging the progression of fatty liver disease. So hepatologists, they would usually start caring about you if you are having the high-end fibrosis like stage 3 or 4. But of course, for public health, it will be very important to know already earlier where you are heading, because that will also link to the potential diabetes and cardiovascular disease. So what do we know about lipids in fatty liver? So, of course, by definition, fatty liver the lipids are essential. It's defining components of the fatty liver disease. So the liver, of course, is also important metabolically. It's the main source of glucose, and gluconeogenesis for amino acids, lactate, and glycerol. Glycerol is highly important here because this is where the triglycerides are being formed through fatty acids plus glycerol. And fatty acids are in fasting state, are taken up by the liver and then oxidized to produce energy and ketone bodies, or reesterified to form deacylglycerols and triglycerides. So, and then the triglycerides are being stored as lipid droplets and secreted then through VLDLs. So from the liver, what we measure then in the blood are then the VLDLs, and the lipids, triglycerides that we measure in the blood by lipidomics are mainly those that are stored in VLDLs. So in postprandially, then we have the novel lipogenesis that is stimulated by carbohydrates and insulin, and what is actually known about in fatty liver is that more fat you have in the liver, also more stimulated the novel lipogenesis is. So it's a bit absurd, but that is one of the things that we know about fatty liver disease, that actually fatty liver will in the postprandial state stimulate more lipogenesis. So synthesis of the fatty acids like myristate and palmitate, and which will then be incorporated into the triglycerides. So I will skip that. I think you all know about lipidomics. So we have many methods, shotgun, and that's what we do. What we do mostly is the reverse phase LCMS, and then we have, of course, our targeted methods. So in our fatty liver studies, we have mostly used reverse phase based lipidomics, and I think shotgun used to be popular at some point, but not anymore. Currently, I think reverse phase is getting back to being recognized as being the very reliable method for lipidomics. So we have done some work already years ago looking at the lipids in the liver. So here, what the goal was basically to look at the serum and see if there is any way from the serum to see, to recognize amount of fat in the liver. So this was not, the goal was not really to look at the staging of the fatty liver at that time. At that time, I think nobody was really looking into that, but more about, can we actually see or predict the amount of fat in the liver by looking at the blood-based measures. So we have the cohort from Finland and then also from Belgium. About half were characterized using biopsy, and another half was using magnetic resonance spectroscopy. And already with 3 lipids, we were able to have a reasonable model. This is actually validation cohort, so this is not discovery. So that was independent validation, but it was not really good enough for clinical practice. But I think it was promising. But was main finding here of relevance is that the key lipids associated with fatty liver disease are triglycerides, which have low carbon number and double bond count. So that was, I think, the main finding. And just going back to that, so triglycerides, they are having 3 fatty acids esterified. So these fatty acids may be polyunsaturated or saturated. So, but basically those that have more saturated and more shorter, like a palmitate or myristate, they're the ones that are being enriched in fatty liver disease. So these are the fatty acids that have been found earlier associated with insulin resistance, and they're also associated with the novel lipogenesis, meaning that they are synthesized in the liver. So particularly like myristate and palmitate, they are not mainly not coming from the diet, but they are coming from the actual the novel lipogenesis in the liver. So that is the kind of lipids that are being enriched and esterified then into triglycerides, and that's what we see also in the lipidomics in blood being excreted by VLDL particles. And there was also independently found in the Framingham cohort that these lipids are also associated with the risk of type 2 diabetes. So then what we try to elucidate is, are these lipids, what is the source of these lipids? So what is this metabolic effect, or is this genetic effect? So we had a study of 125 individuals who were at various stages of fatty liver disease, and we had liver biopsies from all of them. So 125 individuals, and we did the liver lipidomics, and of these we divided them in 2 ways. One is based on the HOMA index, meaning the insulin resistance, so it's basically higher or lower. And then the other way was based on the genotype. So there is a certain common variant in fatty liver, which is called PNPLA3 gene, so-called M variant, which is associated with high liver fat. It's a common variant, meaning it's not harmful, obviously, but about 40% of individuals overall in the world have this variant, but it's clearly associated with high liver fat. So you can see here from the data that we have basically, actually I will show it here. So we have metabolic fatty liver based on insulin resistance, we have higher liver fat, and then we have also genetic fatty liver. So divided the same group in two different ways, either metabolically or based on genetics. So, and in both cases, you see there is a high liver fat, genetic or metabolic. There is no difference in BMI. You can see that insulin, by definition, of course, the HOMA-IR insulin resistant group had higher insulin. On the other hand, there is no difference in PNPLA3, genetic fatty liver. So basically, genetic fatty liver is not linked to the insulin resistance. And then we did lipidomics of fatty liver biopsies, and we clustered the data. So there is no, I will not go into details, but we had 11 clusters of lipidomics from fatty liver. And then we found that in the genetic fatty liver, we had only 4 clusters that were increased, and all of these clusters were basically triglycerides containing monounsaturated fatty acids or polyunsaturated fatty acids. So basically, what we saw that in genetic fatty liver, you have a storage of triglycerides in the lipid droplets, and that was basically it. There was no other component to it. So basically, what that means is that likely the lipids having these variants are being stored, but they are not being there is inhibited lipolysis. So there is the lipids are just getting more stored in the lipid droplets. So that's also actually confirmed from other studies by mouse models, etc. So that is basically what PNPLA3 is doing, and that's what you get higher liver fat, but it is not harmful effect per se to lead to more complicated, more severe conditions of fatty liver. On the other hand, if you look at the insulin resistant fatty liver, you will have also triglycerides stored, but on top of that, you will have 2 other clusters that are linked to fatty liver. One is ceramides, and then deacylglycerols and triacylglycerols containing saturated fatty acids. De novo lipogenesis again. So basically, in the metabolic fatty liver, you will start ending up with lipotoxic lipids like ceramides and deacylglycerols that are actually the drivers of the insulin resistance and inflammation, etc. So obviously, even if you have the same amount of fatty liver, it's not the same. The actual lipids that are there are of course important. So one is inert, and the other ones may be having leading to metabolic complications or immune response. So we have actually since then done a lot of work on the various type of gene variants that are currently common gene variants. So these are not a rare variants characterizing the fatty liver, and actually some of them are being used also currently as drug targets for the NASH. So this is obviously showing that actually lipidomics is a very good way to characterize the fatty liver disease. But of course, it's obvious because fatty liver is, by definition, of course, a problem of the lipids that you have in the liver. So following that, what we did also was looking at fatty liver disease in progressive stages, meaning that not only looking at how much fat you have and characterize that, but also look, is there a way to actually look at the staging of the fatty liver disease, like the different stages of fibrosis or NASH. So there is a European NAFLD registry that was created through the European project that we were participating. And in this registry, there was a whole spectrum of fatty liver disease. So we could actually look at the different stages of fibrosis, which is the most clinically relevant phenotype of interest to hepatologists. So without going into too much detail, so we actually were able to see that different stages of fibrosis or NASH do have very different profiles, so both lipidomic and metabolomic. I will now just focus on lipidomic. But what is most interesting here is that if you look at, for example, fibrosis, which is particularly stages 2 and 3 are kind of the borderline where you start, hepatologists start to care about it, and also it's also the entry point for the clinical trial. We saw that actually the stage 2 seems to be sort of a threshold where lipid changes start to happen. So this is now, you can see fibrosis 0 to 4. 4 is the cirrhosis, so that's already the terminal stage of the fatty liver. And you can see that actually there is a certain metabolic threshold at around 2 to 3 stage where things clearly change metabolically. And you can actually see it even better if you look at the stages, just looking at it, significant changes between 0 and 1, 1, 2, and then 1, 3, etc. You see there is a jump if you go to the stage 3 fibrosis, so seems to be like a tipping point. And the reason why this is really interesting is because this is exactly the point where currently you want to enroll individuals into clinical trials for like a treatment of NASH. So this fibrosis 2 and 3 is really the most interesting transition that one is looking, and obviously also metabolically seems to be the most relevant as well. We have tried to develop that diagnostic model. Well, not too well. I think that it's an okay model. I think it's not worse than currently accepted models, but basically you would have to go over 0.8 AUC if you wanted to get into clinical practice. There was a very nice paper just few weeks ago in Lancet about various kinds of markers for fatty liver, and basically not a single marker currently being considered for approval by FDA or EMA is actually meeting this 0.8 threshold that would be needed for the clinical practice. So, but it is at least promising, but one has to be realistic that we are not quite there yet with our data. What is, I think, the main issue with biomarkers in fatty liver and probably also in other areas is really heterogeneity of the disease. So I have shown you that we have metabolic fatty liver, and then you have, because of genotype, you may have exactly the same amount of fat in the liver, but of course the underlying mechanisms and also of course the lipid profiles will be completely different. So endpoints may therefore vary across different studies. But what is important, we have done a very comprehensive review of the evidence of the lipids and metabolites across many studies in fatty liver, that results are consistent. So you will, in pretty much any study will show you that you have these triglycerides with the fatty liver associated like a saturated fatty acids and short chain. So I would say that most of the studies will report these. Of course, the issue is that specific panels that people report as being predictive of fatty liver will vary a lot. There is, of course, analytical variation across platforms as well. We are currently working with four labs, including ourselves, that we are in the big European initiative. Unfortunately, we are not yet ready to report the results because we just finished the analysis, but we have actually tried to do different panels that were reported in the literature, and then having 4 labs and just try to see what we get by having different platforms and from what I know is that actually the lipids that we measure compared to like the NIST 1950, the reference material, that we are actually quite okay with the levels, but my feeling is that we'll still be quite different when we get to the actual biomarkers. But I think these sort of studies are important, because if you really want to go to the clinical practice, you need to be sure that you can have a very clearly the results that can be validated across many platforms. So now I would like to go a little bit further in terms of the mechanism. So of course, biomarkers is one thing, but for biomarkers in fatty liver disease, there are a lot of options. So it's not just lipids, there is of course many proteins being considered, and etc., and various type of imaging techniques. So, but I think that lipids and metabolites in general can bring to the field, in addition to potentially biomarkers, is really understanding the actual mechanisms of the progression of fatty liver disease. So one way to do that and address that is by studying the genome scale metabolic models. So genome scale metabolic models are essentially approach to put together the whole biochemistry of the human in tissue specific way, and look at the reactions, metabolites, and enzymes, and generate sort of a giant stoichiometric matrix of metabolites and reactions that then will be assumed to be steady state. Then we also, of course, try to impose certain constraints. Otherwise, of course, it will be impossible to do anything. So basically, the idea here is to look at tissue specific, so which enzymes are being expressed in the liver, for example, but then also gene expression will then give you additional constraint. And here we want to see, based on gene expression, also if there is any difference between the fatty liver or not. So you have to use transcriptomic data as well. So we have done that. So we had a liver biopsy gene expression data from our paper in transcriptomics paper, and then we have lipidomics, metabolomics, done in serum. We didn't have metabolomics, unfortunately, in fatty in liver, because we just didn't have enough samples. So all the sample was used for it was a needle biopsy, so it was not possible to do metabolomics. And then using genome scale metabolic modeling, we were able to then look at the different stages of fatty liver disease. So this is the same population as we had in the metabolomic study, but it was obviously smaller because we it was only about 200 something samples that were done for transcriptomics from the liver biopsies, and then using the models. So we looked at the transcriptomic data from different stages of fatty liver, and using that allowed us to then constrain the metabolic model. So based on the genes that were expressed in different stages of fatty liver disease. So you can see here that, for example, if you look at just different stages of fatty liver disease, these are division in different stages, either NASH, different fibrosis stages, or then combining, and this is more reliable way, more advanced stages versus less advanced stages versus the NASH in more unadvised, so lower stages of fibrosis, for example. So you can see clearly there are certain stages that are highly upregulated in the more advanced stages of fibrosis, and a lot of these include the lipids. So this is not really modeling yet, but here what we saw was, for example, that if you look at the reporter metabolites are basically metabolites where a lot of changes happen around. So if you look at the enzyme expression, and around them specific metabolites, you will see a lot of changes happening at the expression level. That's what you will then report here as being the important reported metabolite. So this is sort of analysis that is common in the genome scale metabolic modeling. And I think in these, what we saw is that there was a clear downregulation of for example, the glycosphingolipids, that is this big cluster here. But of course, there were many others. So we have not gone yet. So we also saw icosanoids being very important. But we have focused particularly on sphingolipids because we know that these are highly important also in the insulin resistant. But I think we could probably follow on a lot more than that. So we saw, for example, that the novel sphingolipid pathway was downregulated in advanced fibrosis. So you can see here reaction 7 and 8. So these are over here. And ceramide production from sphingomyelin was also enhanced. And there was also marked increase in sphingosine recycling pathway. That is the reaction 10 that was highly upregulated in the more advanced fibrosis. So we have currently another work in progress looking at more central metabolic pathways, and clearly what shows that our data that actually the advanced fibrosis seems to have a very different metabolic phenotype, and I think this is something that has not been reported yet, and it's not really understood. And of course, it's very hard to study because you really need to have liver biopsies to get into this point. But at least this study has shown that the sphingolipid metabolism seems to be disrupted already in this progression of fatty liver disease. And we were also able to show that, for example, there are pathway differences because if you look at it, 3 different variants, the most common variants in fatty liver, that is DM6SF and then PMPLS3, as I already mentioned, and HSD17. So they all have different metabolic phenotypes, and you can actually see here that for example, in PMPLS3, if you look at the wild type versus the M variant that is associated with fatty liver, they have clearly different ceramide levels lower in PMPLS3 M variant, and also erythropoietin is lower as well, as was predicted by the genome scale metabolic model. So for the end, I want to mention also the potential role of the environmental contaminants. So this is one area that is getting more and more attention, also in fatty liver, but in general, of course. And the problem, of course, in this field is that it's very hard to understand what is cause and what is effect, because when you study a lot of chemicals and then you have a lot of phenotypes, you will always find some associations. So it's very important also to understand what could be the underlying mechanism. So we were particularly interested in PFAS because they are going to be banned actually overall in Europe. But there is also another reason for why PFAS are interesting is because they are directly interfering with the bile acid metabolism. So they are actually PFAS are using at least PFOS that is most studied, and also PFOA. They are utilizing the same transporters in the gut as bile acids to re-enter the enterohepatic circulation. So they are competing basically with the bile acids to re-enter circulation, and that is actually the reason why they are being so persistent in the body, because they are able to circulate and not being excreted, utilizing the basically bile acid pathways, and that's the reason why I think over 95% of them are re-entering the enterohepatic circulations. So, but PFAS are basically fatty acids, short chain, that are replacing the hydrogen with the fluor. So they are having lipid-like properties, but on the other hand, they are actually somehow managing to behave like bile acids. So they have double double role in the body. And of course, in fatty liver, that is both are very important, fatty acids and bile acids. And for that reason, we were very interested in exposure. And we looked at the same study that we published, also these genetic variants based on liver biopsies. So here we have liver biopsies from the same study, but we also looked at the bile acids both in the liver and the circulation. And then we also looked in circulation the contaminants. We were not able to look at the contaminants in the liver because there just was not enough sample available. But we did study contaminants in serum, particularly PFAS, but also there were some others as well. And we clustered contaminants. So what you see here, EC is basically a cluster, and we named some of them like PFOS and PFOA. You can see that they are clearly associated with NASH, meaning more inflammatory condition of fatty liver. But there is also, interestingly, big difference between the males and females, and actually that was our main finding of the study. And if you look here, this is actually truly striking data that got quite some attention. If you look males and females, and this is just simply correlations between the different PFAS, and then correlation with different lipids. Like these lipids here, triglycerides, they are linked to the fat in the liver. So these are associated then with the NAFLD and NASH. But if you look males and females, you will see that the actual direction is completely opposite. So you will have actually negative correlation, meaning that I wouldn't call it protective, but it's clearly negative in males. But then in females, you will see clearly positive correlation between these lipids and the fatty liver disease. Basically, in females, it looks like what you would expect. In males, it doesn't. So there is clearly a dose dependent, and in this case, exposure was in absolute terms relatively low. So we are talking about a low level of exposure, and what you see here is basically completely opposite effect on the lipids in the liver in relation to exposure. And we were able to actually see that if you look at the mice that was done in Boston, in just PFOA study in the humanized PPAR alpha model, and actually in this case, females were behaving more like the mice were behaving more like females. So in terms of the impact of exposure, but we were able to show that actually there was a similar effect than of exposure in mice, male or female, as it was found in females in human study. So in conclusion, what we learned in these studies is that that NAFLD is very heterogeneous. It's not only about fat and amount of the fat, but it's also about quality, meaning the actual lipids that you have in the liver is very important. And lipidomics has, in our studies, and we have shown that basically it's a very important tool to unravel the heterogeneity of the fatty liver disease. So we have found that there are certain lipids and metabolites that vary with progressive stages of fatty liver across the full spectrum of disease. So that there may be biomarkers, and particularly we are very happy to see that actually this fibrosis transition from 2 to 3, it seems to be also metabolically a critical tipping point. But as I showed here, it seems that currently with the diagnostic tools that we have, we are not quite there yet. So there is a lot of effort, of course, by from many sites, omics sites, but currently there is nothing yet available that would really replace the biopsy as a kind of endpoint, the hard diagnostic gold standard. So it's just being realistic about that. But on the other hand, genome scale metabolic models has give us some insights into the fatty liver disease staging and pathways to new therapies. And what we have found, particularly, it seems that environmental chemicals seem to be, number one, they are sexually dimorphic. So there is obviously different effect in males and females, which we do not understand. We now done steroids also, and we see clear dominance and the steroids, but I think we are not yet understanding the mechanism behind these sexual dimorphs. But it seems that the main interplay, I mean, the way the PFAS are acting on the fatty liver disease, maybe through the bile acids, because of course bile acids are competing with PFAS in terms of the enterohepatic circulation, and bile acids are of course very important also in the steroid metabolism. So there is something that we are currently looking into. So I think this is more or less yes. So I'm finished here, and I'm very happy to answer any question. I noticed now that there are quite a few questions. Do you want me to go to the questions, or should you just ask me?
I will read them to you because I've kind of
I have noticed quite a few questions came through this.
Yeah, no worries. I've grouped them into categories. Great talk, very interesting. Always nice to learn about a little bit more about biology and new topics. So just starting off with sample type and maybe your experience in sample prep. Is serum better than plasma for non-alcoholic fatty liver disease analysis? Would lipoprotein fractionation add to the picture to improve the area under the curve for, diagnosis?
Yeah, so generally, I think it's advised to use plasma, but we used serum as well. So I think it's both have worked, but certain metabolites are better in plasma for sure. Generally, I think people would recommend plasma EDTA to be used for lipidomics, for example. For metabolites, I think it's about the same. You don't want to use citrate, of course, polar metabolites. So, but I would say generally plasma is recommended, but I think many of the studies we did here were done in serum. So there have been a lot of studies comparing serum versus plasma, various kind of plasma and various kind of procedures. Of course, how quickly you freeze the samples and how they're treated. There is room temperature. Of course, with serum, you don't want to have too much room temperature exposure, so that's a problem. So I think these are, of course, all the considerations that go into this. But lipoprotein fractions, I think that will be absolutely very important. So we have this epilipid network that is having lectures, the meetings every month. And actually in May, we will have a lecture exactly about that. So whether HDL is actually highly informative for fatty liver disease. So yes, I think, of course, the problem with having fractions is that they are very tedious to acquire, right? So you have to do the ultracentrifugation, etc. But yes, absolutely, because what we measure in blood by lipidomics is essentially bulk profile of everything that is there. So VLDL, LDL, HDL, and whatever else. So ideally, you would separate these into different fractions because that's where the lipids are, and each lipoprotein will have its own functional role. So yes, absolutely, if one could do that, it's not for diagnostics, it's probably too tedious, but for mechanisms, having the fractions of lipoprotein would be very informative.
Yeah, but you could also have specific antibodies for specific lipoproteins, tag them, on beads, fractionate them. Seriously, shouldn't be now better these days?
Yeah, I agree. I think that we have actually done the systematic study of different lipoproteins in fatty liver. So it was a long time ago, and there have been a lot of effort since then. The hard gold standard for lipoprotein separation is still the ultracentrifugation, and that can take a long time. But yes, there have been a lot of novel methods. And I agree that probably one should revisit that to really look at different lipoproteins, even if it's not perfect, still would be very informative.
The next question we have is about animal models. What is the role of animal models in discerning mechanisms towards different stages of fatty liver disease?
Yeah, I think this is actually a very good question, because that's where the drug companies are really interested, given that there are many of them are running trials and have drug programs for the NASH. So there is this project that we had. I will just mention it here because so that's this litmus. So that's a very big project, innovative medicine initiatives that has pretty much all the major pharma companies involved, like Pfizer and Takeda and Novo Nordisk and I think it's like 9 major pharma companies that are running the NASH programs right now, and they all are very interested in development of models. So basically within the litmus model, and that's a group of Antonio Vidal-Puig from Cambridge. So he has actually done the developed models that were, because we have so much human data in this project, to correlate that with the human data. So he has actually developed the models within this project. It's not yet published, it's just submitted from what I know. That actually is it's a model that defines different stages of fibrosis and that correlate very well then with the human phenotypes. So, but it is clearly, as of now, unmet need, because currently the models are based just like on diet, and then they develop obesity, and because of that they develop fatty liver, but it's not really defining the actual trajectory of progression toward fibrosis. So there is, it is very complicated, and I think that currently in this litmus project they have developed the new models that will actually correlate very well with the human progression of fibrosis. So, but it's not published yet.
Okay. The next question is on some of the data that you had shown from your iScience paper, which I believe came out in 2022.
Yeah.
The most differently regulated metabolic pathways were neither on TAG metabolism nor glucose metabolism, but sphingomyelins and PI lipids. How do you interpret that finding?
When we get to glucose, we are just putting together another paper on that because it seems to be more complicated than we thought. But sphingomyelins, I think, are easier to interpret because I mean, there are different ways to, and ceramides are at the center of it. So we were able to show that ceramides are increased with progressive fatty liver. But what we were surprised about is that this was not because of the de novo synthesis of ceramides that we would expect, because when you have saturated fatty acids like palmitate, this is a precursor of ceramides. But these reactions were not increased, but it was the actual, what was changed was really the incorporation of ceramides into the glycosphingolipids. So ceramides were accumulated because these pathways were downregulated. So that I mean that's interpretation. So we were not going here in this study into the glucose metabolism, but yeah, there were actually many pathways that were perturbed. We were specifically focusing on the lipids because we had lipidomics data to validate the findings. So we were able to verify predictions from metabolic models, but we have since then also looked at the polar metabolites, and that's actually just being prepared right now and validated in other studies because we didn't have enough data from the study. But I think sphingolipids, of course, are very important. They have been reported being increased, ceramides in obesity, insulin resistance. So that seems to be in line with what we have been finding here. But again, the main surprising finding here was that it was not because of de novo synthesis of ceramides, but it was more about lack of incorporation of ceramides into the glycosphingolipids in the advanced stages of fibrosis.
Okay. So I think one of the interesting parts of your presentation was, for me, the exposure story linked to fatty liver disease, specifically highlighting PFAS and PFOS. But there was a little bit of, maybe if you could clearly explain the possible mechanism of how PFOS interferes with bile acid transporters, because bile acid concentrations are really huge, whereas PFOS concentrations might be very low. So how do you view that? Like, how is that mechanism explained? If you could further explain that.
Yeah, I mean, there are many ways that the PFAS can contribute to the fatty liver disease. I mean, we have had a review about that, but the main thing in terms of, it's not directly competing, because yes, you're right about the concentrations, but particular PFOS and PFOA, of course, they are at high concentration and they are found pretty much in everyone, every human. They are utilizing the same transporters as bile acids. So they are actually not maybe directly having a major influence on the bile acid metabolism because of concentration difference, but they are, for the reason of utilizing these transporters, they are able to be persistent in the body. They are also, there was a question about FXR. Yes, they actually are having, that's not in this study, but we have done actually in study that is now under review, looking at the PFAS and PFOA in the human fetus. And we found that actually the major impact of PFAS and PFOA in the fetus is actually FXR, seems to be most correlated gene. I mean, it's like really manifold upregulated with higher exposure to PFAS and PFOA of maternal exposure. So yes, these key genes in regulating the bile acid metabolism seem to be affected by the PFAS. So that much we know. But you're right that having the same transporters is probably not explaining everything, because there is a clear concentration difference. I mean, we do have now many new conjugates that we are of course very interested as well from the gut that were reported by Peter Dorenstein. So and we see them in the stool samples and also in the liver biopsies. So I would say it's very complicated, but I think that PFAS are clearly interfering with the bile acid metabolism.
I'm not familiar with.
That will also mean that they are interfering with the steroids. So, but the exact mechanism is probably more complicated than we think. So there is transporters is one thing, but there is of course also the key genes. I mean like FXR that are also impacted by the PFAS. But there are probably many other ways. So it's not just a single pathways that are being affected.
I was going to ask, I'm not familiar with FXR. What does that stand for?
Farnesyl X receptor. So that is actually the key receptor for bile acids.
Oh, I see.
In the liver, yeah. But it's also in the gut. So yeah. So it is the most studied, I would say, gene in the context of bile acid metabolism.
Great, thank you. How many PFAS or PFOS are detected in fatty liver disease samples that you've run? Are we talking about like hundreds or thousands?
No, So this was more like a semi-quantitative, so quantitative, mainly quantitative. So it was, I think that totally about 20 contaminants. But yeah, that's a good point. So we did not run a very untargeted approach here.
And you were able to find 20 standards for those?
Yes.
Those?
Yeah,
Okay.
we do have quantitative method for PFAS, yes.
Oh, that's
So,
great.
but I think we do currently, not at that time, but we usually run about 30 PFAS as quantitative, and then many more than as semi-quantitative. Yeah.
Do you plan to screen for other environmental chemicals that impact fatty liver disease in the future?
We do. I mean, we actually, even in that study, we were not solely focusing on PFAS. It's just that PFAS ended up being the most interesting. But yes, we are having a very comprehensive methods that can cover partly quantitative and then partly semi-quantitative, like suspect screening. So yes, we are not solely focused on PFAS. Of course, PFAS is easy to detect, the easiest ones. But yeah, we are looking at others as well.
Yeah, they're just about everywhere.
Yeah,
that's for sure.
Exactly. I mean, they're going to be banned soon in Europe, but of course they will not go away for next few decades. So
no, I wouldn't imagine even in our lifetime. So there are some general questions we'd love to hear your opinion about. How important is it to go towards absolute quant in lipidomics to better compare findings between studies?
Well, I think this is a very sensitive point. And opinions differ because the way people define absolute quantitation is also variable, and I strongly disagree with many opinions here. I think it is important. I mean, the state of the art, just first to understand. So what we can do is we have for example, class specific standards, and then we can calibrate against those. So we have a fairly, I would say, reliable way of quantifying, but it's not obviously absolutely quantification. So if you really want to do absolute quantification, you would have to have standards for everything, and that's not really realistic. I mean, unless you were like labeling the whole mouse or something, then you will have a C13 mouse. They would not survive to begin with. It's not possible really. So I would say that it is important to be as accurate as possible, but you also should be realistic about how far you can go. So I mean, in our studies in polar metabolites, we usually have like certain number of metabolites up to 100, where we have standards, and we have those in the sample. So we quantify a certain amount, and then the rest is then semi-quantified. And in lipidomics, you have the class-specific standards, and then you do that in reverse phase, where we are very much addicted to reverse phase in lipidomics. Shotgun people will, of course, highly disagree. And then now they are going into helic, but helic is not very good, like for neutral lipids. So at the end, I think the most important thing is that you are being able to report what you are doing and have as many standards that is feasible. But I don't think that absolute quantitation really, I mean, it has been used, the term, in lipidomics, but it was, I would say, abused, because it's not truly, in most cases, it's not. I mean, in bile acids, yes, you can quantify all of them because we have standards, but like in broad lipidomics assay, I don't think you can quantify everything. So that's my view. But you try to be as accurate as possible. I think that's pretty much the goal that you have in lipidomics. I mean, Oliver, I don't know what you think, but my feeling is that we are not really in a realm of absolute quantitation.
Yeah, well, obviously the terms are also confusing, because then people say semi-quantitative, and what is semi-quantitative meaning is that it's we don't have the words. There's different strategies, but obviously that's a big discussion, and how to make metabolomics and lipidomics more standardized, more comparable between studies, right?
Yeah.
Even the naming, I saw that you had names for lipids that sometimes non-standard, sometimes, because there's not easy to name those. And, you had also lipids that were like this or that lipids. And how do you then?
Yeah, I mean, well, one is of course nomenclature. The other thing is the comparability. So I think when you go to comparability across experiments, I think that it's very important to use like a standard samples like the SRM 1950 NIST sample. I mean, we use that in every study, and I think it's very important to be able to. We are now, more and more projects that we are involved in are also involving studies where metabolomics was done by somebody else, like Metabolon or whatever. And then, of course, you don't want to rerun yourself with your own method. You want to be able to then match the data that they had already from before with what we are generating in the new samples. So there, I think it will be really essential in these cases to have sort of reference samples that everybody's running at, like SRM 1950. I think this is what we do. And I think more and more people are starting to use those, because I think this is the best way to match the studies across, I would say, absolute quantitation. I know that in type 1 diabetes, I didn't talk about it, but in type 1 diabetes, for example, the big discussion is about antibody titers, so autoantibodies like insulin, GADA, and whatever. And they have quantitative assays developed over like 30 years. And they have run many ring studies across samples, and they have validated methods, reliable, quantitative, and yet still you will see huge variability across these assays. So even if you claim in metabolomics that you have a quantitative method, you will see that your method will vary quite a lot with another method as well. So I think that having the reference samples like NIST standard, I think it's probably the most reliable way of comparing the studies.
Okay.
Great. And one of the last questions is, in general, how specific are the candidate biomarkers to fatty liver disease against other liver metabolic diseases, if not found by designed study or a given experiment, but what's reported in literature?
I think specificity is, I think that is, of course, a big problem that generally, but we find in epidomics, it's very much correlates with the risk also of type 2 diabetes. So it's not specific to fatty liver progression, but it's more linked to the insulin resistance. There are markers that are currently considered in NASH, protein markers that are quite specific to fibrosis development. So it does depend on case by case what it is. So I would say metabolic markers that we have so far are mainly linked to the insulin resistance. There are protein markers that are more specific to fibrosis, but I would say a combination of those would probably be the best option for clinical utilization, because obviously lipids are informative, but proteins as well, but they are complementary. So, but I would say that there has not been yet a really comprehensive study looking at various kinds of liver conditions, not just the non-alcoholic fatty liver. There was one very nice paper just some months ago in journal, it was in hepatology, showing that alcoholic fatty liver actually has a very different lipid profile than the non-alcoholic fatty liver. So it has more of a inflammatory signature compared to the metabolic fatty liver. So lipidomics looks quite different, very different in these two conditions.
Great. I mean, I would like to open it up. There's more time to ask questions. If anybody would like to unmute and ask Dr. Orešič any questions about fatty liver disease or lipidomics, exposure related studies. Am I frozen? No.
No, it's okay.
Okay.
By the
Everybody
way,
was so,
next year will be 20 years of MZ Mine, so there will be apparently a party. So I talked to Tomasz Pluskal, who is now really running this, and there will be probably sometime in spring. There will be a meeting in Prague dedicated to MZ Mine.
yeah, that's a
interested.
big you know, time and point for
20 years is a
software.
long time.
Yeah, 20 years is a long time. Yeah, you had mentioned 2 other events as well.
Yeah, there's Osaka, of course, the main conference, Metabolomics next year, and then there is going to be Nordic conference as well in the autumn next year. Yeah, but I think the main MZ Mine event will be independent from this event.
Great.
All right. Well, thanks everyone for joining. If there are no further questions, Arpana, do you want to introduce our next speaker?
Yes, I'd love to. So just one second here. Our next speaker is going to be Dr. Susan Bird from Thermo Fisher. She's the senior manager for metabolomics division, and she's actually going to be talking about the conversation that we were just discussing in regards to, you know, targeted or quantification, semi-quant in their new technology called SQUAD. Title is called Metabolomic Squad Goals, Orbitrap Workflows to Empower Your Research. And I don't remember what SQUAD stands for, so maybe Oliver, if you
simultaneous quantification and untargeted discovery.
Thank you. Yeah, so essentially it's, you know, doing untargeted and targeted metabolomics in one given run. So we're very excited to hear about this new, you know, methodology from Thermo, and they're going to be talking about it at ASMS as well. So we're going to get hopefully a little bit of spoiler reveal before ASMS.
Right.
Hopefully everybody can join. Great. Thank you.
Thank you so much, Matej, for making time on a, you know, Friday evening.
Yes. Thank you.
Okay.
We
A
appreciate
pleasure.
that.
And I
have a great
hopefully
weekend.
see some of you in Niagara, as I said.
Bye-bye.
I will be there. Okay, bye-bye.
Okay, bye-bye.